Short-term inflammation is beneficial long-term. When that sets in, some physician someplace will say, now arthritis has set in. All right, so now we are defining fairly carefully that the body must have a mechanism to allow short-term beneficial inflammation and terminate long-term destructive inflammation for maintenance of health. You folks are young. You have robust bodies. Your bodies are working near perfectly. Our bodies, well, we're sort of like antique cars, kind of shine on the outside, but the parts are wearing out on the inside. All right. Now, let's go back and revisit cortisol. What else? How could the body have been designed to accomplish the objective of allowing short-term inflammation that's beneficial? Our conclusion in the 70s, in the early 80s, is that there has to be a cortisol pulse, represented by that large spike on the screen. So that was put in as a hypothesis. A hypothesis is a $25 word for guess. But when you're working in science, you use hypothesis, not guesses, as the word to describe it. Well, now, if that were there, this is how your bodies are working without arthritis, then this must be how Helen's body is working with arthritis. So when the good inflammation comes, and after a little while, then the body says, rip it up, put a lot of cortisol in the body, seal the leaky holes, let the swelling go down, stop the immune response, and allow healing. But Helen's body can't do it. So her body tries to stop it, tries to make the cortisol pulse, but it is no longer able to make it in sufficient concentrations to seal the leaky blood vessels, to stop the immune response, to stop the swelling, to stop the pain, and all of those things. Hypothesis again. If that is the case, then this is the solution. You teach her to take cortisol tablets and manually build it up again. Replace the missing by teaching her when and how much to take. Seal the leaky blood vessels, then the swelling goes down, the pain goes down, and then don't take it every day, just then. It's a switch. The amount in the bloodstream all the time has nothing to do with rheumatoid arthritis. Well, if she just takes it now and then to when she hurts, in other words, take it on the bad days and not on the good days, she uses so little that she never gets the cortisone side effects for two reasons. She's under the safe limit, and secondly, she's just replacing the missing that should have been there anyhow. So for 22 years, then Helen has had no side effects and she remains pain-free by doing this. You can't say anything about that? You know, arthritis has plagued us for thousands of years. Even the dinosaurs had had it. Now I'm telling you in like 3.5 hours how it works. You don't believe me. That's why you won't say, wow. Huh? Okay. Now, that's true because you don't solve a disease by one patient. Good. So then we need experiments, but there's only a limited amount of experiments that we can perform on human beings. We'd like to do more, but humanely we can't. So we have our friendly animals around. By hypophysectomy, that is, going into the brain and keeping the hypothalamic axis, or so that the hypothalamus cannot signal the adrenal axis properly. By that surgery, then we are assured that the cortisol pulse that we postulated would not be made, could not be made by those rats. If that were the case, then if we inject these rats with a inflammation-inducing agent, in this case carrageenan, a standard in the field, then it should swell more intensely and for longer periods than it would be in a rat that didn't have hypophysectomy, and that's true. We published that. So this is, we could make then, in lay terms, rats appear to be arthritic by simply exposing them to hypophysectomy. Experiment number 1. Arthritic rats made normal. So we took the hypophysectomized rats that couldn't handle inflammation well, and then I taught the students that they should use the Helen plan on them. The Helen plan is this: to give them sufficient amount of cortisol to the point where they run around their cages like normal, like everybody else's rats in the animal laboratory. Stop and watch. Well, since these are hypophysectomized rats, we had to induce arthritis in them again. So we injected carrageenan to create the inflammation. The inflammation caused them to squeal and be pained, and then the students would take the cortisol and bring them back pain-free in very short order. And they could do that repeatedly. So our rats really behaved like normal rats. So we generalized and we published that. All right, now the folks at the National Institutes of Health and said, all right, Stenberg, you have this fancy idea about the cortisol pulse. We have the Lewis strain of rats that always develops arthritis right after birth. We think the answer to arthritis lies within this strain. If you're correct, then these rats were born without the ability to make the cortisol pulse. In other words, there's a genetic defect within them that disallows them to make the cortisol pulse. They looked, and that was true. Now we're talking the third line of evidence. The fourth was the physicians in London said, okay, Stenberg, you have this fancy idea about the cortisol pulse and its role in arthritis, inflammation, rheumatoid arthritis. Suppose we take another experiment. This is one that I dreamed we could do, but I didn't know how to do it, and these ingenious people came up with it. They said, well, let's take a group of people that don't have arthritis. I hesitate to call people normal because I don't know if we have any normal people. We have people without disease, but we're all screwy in some way or another. All right, these without any arthritis, and then we perform a surgery on them, necessary of course, and then watch for the cortisol pulse after the surgery, because a surgery is like an injury. An injury is one of the big four things that cause inflammation: stress, injury, allergy, and infection. All right, when the surgery cuts, they create an injury. After the surgery comes the cortisol pulse. That's well known. I said, fine. Now we have a second group. We're going to take people then with chronic arthritis, chronic rheumatoid arthritis, and have them undergo the same surgery. Of course, it has to be those where it's necessitated. But when we get enough of these people, let's compare the two. Those with chronic rheumatoid arthritis, those without it, same surgery. Then the people with chronic rheumatoid arthritis will not have the cortisol pulse after surgery, if I'm correct, and I said that's what I would guess. They looked, and that was true. Then our division chief of rheumatology said, okay, let's do a double-blind crossover clinical trial. For you budding scientists, let me define what that is. The physician doesn't know whether the patient has got the active drug or the placebo. The patient doesn't know whether the patient is getting the active drug or the placebo. That's a double blind. Both the attending physician and the patient doesn't know, so they don't introduce a psychological factor into the clinical trial. Then we put in a second feature which makes it